So, good morning again. Did I told you about the sigma factor, about the RNA polymerase in prokaryotes? Okay, so this is transcription, Fatima. And did I told you about this? Okay. I told you this part, and this part too, yes? Okay. So this is transcription in prokaryote. So I finished a transcription in prokaryote for you. Am I right? So this session, I want to talk about translation in prokaryotes. We want to talk about translation in prokaryotes. We did not explain it. Explain this picture, you mean? I didn't explain this picture. This. Okay, I will tell you. Okay, no problem. Okay. After this, I want to talk about translation. So up to now, listen to me. We talked about prokaryote replication. After that, we call about prokaryote transcription. And now, in this session, we want to talk about prokaryote translation. Okay? So let's explain this picture too. Look at this. I told you, listen this please. I told you we have holoenzymes. Do you remember the particle of holoenzymes? Do you remember the particles? The particles of holoenzyme was, can you tell me, alpha 2, beta prime, omega, and sigma. Remember? Okay, I told you which part can recognize the promoter. Can you tell me, please, which part can recognize the promoter? Which particle, which part of the holoenzyme can recognize the promoter? Sigma factor, Fatima. Now, look at here, please. Look at here. I told you we have sigma factor in the RNA polymerase, which can recognize the promoter. Okay, remember? So look at here. We have core enzyme alpha 2, beta prime, omega, and after that, I told you if we add sigma factor, we call it holoenzyme. What is the function of sigma factor? Sigma factor is the part which can recognize the promoter. So look at here. This is RNA polymerase, and this blue one. Can you see the blue one? This blue side is sigma factor. Sigma factor can recognize the promoter. We have 1, -10, and -35 region. Yes? And where does the transcription start? Where does the transcription start? Transcription start here. +1. Okay? So look at this picture. Look at this picture. I told you we have holoenzyme. It can find the promoter by the sigma factor. After that, I told you by the change of its structure, the closed DNA can change to open DNA, so the DNA will open. No problem up to now. So change the structure of RNA polymerase and it can change it to open. And after that, the primer synthesize. I told you we should have 7 to 12 nucleotide of primers. If we have less than these nucleotides, we have abortive initiation. I told you if there is less than 7 nucleotides, the transcription doesn't start. After that, I told you sigma factor dissociate, and I told you which protein comes. Can you tell me which proteins come? NOS A come and elongation continue. Do you remember up to now? Do you have any problem? So whenever think you have any problem, please ask me. Okay? So no one has problem here? So very good. I just want to talk about the termination of transcription in prokaryote. We want to talk about termination of transcription in prokaryote. So I told you we can have two different termination. One is row dependent, and one is row independent. Row independent or intrinsic. I told you, tell me about the translation in Arabic. Do you know intrinsic in Arabic? What does it mean? Zat. Yes? Am I right? Zat. Zati. No, intrinsic. I mean, there is something in the DNA. Intrinsic, it means zat. Am I right? Zati. حقيقي. Okay, thank you. So, I didn't explain it. I didn't explain it in the previous session. So look at this picture. Now I just want to talk about row dependent. Look at me. No, let's talk about row independent. In the row independent, we have, for example, this is our RNA. At the end of the RNA, we have one part of GC palindromic region. We have GC palindromic region. And after that, we have A rich region. So look at me, look at this. What will happen if we have transcription? I told you if RNA is synthesized from the DNA, which is GC rich palindromic, what do we have in the structure of RNA? Do you remember? I told you we have hairpin. Did I explain about GC palindromic? As I told you, in the region which is GC palindromic, when RNA synthesized from DNA, we have hairpin structure. We have hairpin structure. So, if we have hairpin structure, how is the strength of RNA and DNA? How is the strength of the bond of DNA and RNA? The strength is weak. Yes? And after that, we have U and A. How is the strength of the bond? U and A, the strength is weak too. Okay? So, in the end of some genes, we have GC-rich palindromic region, and after that, we have A-rich region. So, after the RNA is synthesized from this region, because this region is GC palindromic, we have hairpin in the structure of RNA. So, the strength of the bond is so weak, and after that we have the region poly-U, the strength is weak too. So, NOS A. So, NOS A. Proteins come here, and it will help to finish the transcription. So, look at this picture. This is GC-rich palindromic here, and after that, the RNA which synthesized from this region will form hairpin structure. NOS A attached to this hairpin. Look at this picture. NOS A attached to this hairpin, and after that, we have A, and the RNA is U. So the strength of the bond of the DNA is so weak, so the RNA is separate from DNA. Understand? Anybody? Anybody understand? Okay. I told you. What about the TC? What do you mean TC? Where is the TC? Where is the TC, Fatima? When a start part TC semen organic. No. Look at here. It's the structure of this is the structure in the end of the genes. Okay, we have GC palindromic region. We have G and C palindromic region here. Okay, and after that we have A-rich. We have A. This is the structure of termination. This is the structure of end of our gene. End of our some prokaryotic genes. Okay, so we have A here. The RNA is U. So the strength is so weak and they separate. We have T and C in the before, you know. Look at here. before we have every nucleotides. But in this region, at the end of the gene, we have one GC palindromic region, and after that A region, A rich region. Okay? This is just the end of the gene. Thank you, Fatima. So let's talk about row dependent. Let's talk about row dependent, the first one. I told you, listen to me carefully. I told you at the end of some genes have GC rich palindromic and A rich, but some of the prokaryotic genes doesn't have GC rich palindromic and AT. So how transcription end in this kind of genes? Understand? I told you some of the genes has this GC palindromic and AT rich region, so the transcription can end. But some of the gene doesn't have this region, so we have raw protein. We have raw protein, which is hexamer. What does hexamer mean? Which is hexamer. What does hexamer mean? And it is helicase. Hexamer means it has six different parts. Yes? Okay. Has six different parts. And what does helicase mean? What's the function of helicase? What's the function of helicase? Open the hydrogen bond. Open hydrogen bond. So it is helicase and it needs energy, which means it is GTPase. GTPase. I told you, when the protein is GTPase, it means it hydrolyzes the GTP, which means it needs energy. So look at me. Rho, I told you, is a hexameric protein, which is helicase, and GTPase, it's attached to a region which is C rich region. So at the RNA which is synthesized newly, we have one region which is C region. So in our RNA, we have one region which is C rich region. This is the signal where Rho attach to this region. We call it root. Look at here, this green, we call it root. Rho attached to this region because Rho is helicase. It can separate RNA from DNA by the use of energy. Which energy? GTP. So I will repeat again. Listen to me. I told you, at the end of some prokaryotic genes, we don't have GC rich palindromic, and after that A rich. So intrinsic can't happen. So we need another protein which is Rho. Rho is a protein which is hexamer, which is helicase and needs energy, which means it is GTPase. Rho attached to one specific region in RNA, which is C rich, and by the function of by the use of the energy and by the function of helicase can separate RNA from DNA, and the transcription will end. Understand? Okay, so thank you so much. Thank you so much. Do you have any question? Do you have any question here? Did you understand? Do you have any question? Do you hear my voice? Do you want me to repeat again this part? Okay, no problem, Roya. Just listen to me, Roya. Anybody, listen, please. I told you, do you understand this part? It was row independent. Okay, we don't have row here. By the structure of the gene, the termination will happen. Okay, what is the structure of the genes? I told you we have GC palindromic, and after that A-rich RNA form hairpin because the structure of GC palindromic, and after that we have UUU. The hydrogen bond strength is so weak, so RNA separate from DNA. Okay, this is the first part. Now we want to talk about the second part. This is so simple. Just listen. We don't have this structure. We don't have GC palindromic and A-rich in these genes. Okay, so we use one specific protein, which is Rho. What does Rho do? The Rho is a helicase. So when it is a helicase, what does it do? It can separate the hydrogen bond. Yes, we have RNA and DNA hydrogen bond. Yes, so Rho is a helicase and it is GTPase attached to a region in RNA which is C-rich region. So we have one specific region in our RNA. It is C-rich and Rho attached to RNA, and because it is helicase, it can separate RNA from DNA, and the termination will happen. Does it need energy? Does it need energy? Yes, it needs energy. Which kind of energy it needs? GTP. Okay. Do you understand? Thank you so much. Thank you so much. I think if you want, we can have 10-minute rest, and after that we can start translation of prokaryotes. Okay, just have 10 minutes rest and come back. Okay, hello again. Did you had some rest? I see. You want the previous lecture. I'm so sorry, I had lots of work to do, so I couldn't send you. Okay, no problem. I will send you. I will send you up tomorrow. Up to tomorrow, I will send you the last session. But here is translation. You can download it from here. You can download it, the translation lecture from here, and replication and the transcription. I will send it to you. Don't worry. Up to tomorrow, I will send it to you. Okay. All of you download the prokaryote translation from here. Okay. Here you can download it from here. This side you can download it here. Right at the end. You can see my mouse. Okay. Did you get it? Did you get the file? This is about translation. I know. I told you this is translation, Merve. Okay. Replication and transcription, I will send it to you. So, look at here. I just want to talk about translation. I just want to talk about translation in prokaryotes. Okay, so I told you, let's get the picture. Look at the picture. We have three different parts in transcription. Let me write it to you. One is the first one is initiation. The second one is elongation.